I don't think you will get a question where you will have to use Excel during the exam. I'm pretty sure whatever values will be required, they'll give it to you if they're giving you a question like this. Don't worry about it. I'm just showing it so that in case you need it in your research or somewhere you're doing calculations, it'll be easier for you. Okay, I think we can start right now. I hope my PPT is visible and I hope I'm audible to everyone, right?
Yes, ma'am.
Okay. Yeah. So, in case there are people who were not there last week, I'll just give a small introduction first. So, hi everyone, I'm Anakha, and I'll be helping you solve a few sample problems that are sort of related to each week's assignment that you get. It won't be the same questions, they'll be loosely based on them so that you'll have a easier time solving your own assignments. Okay, I can't discuss the assignments with you. If you have doubts, you can ask me. Maybe I can help out without telling you what the answer is. And these problem solving sessions will happen for all 4 weeks. So this is the second session, and we'll have two more till the course is over. Okay. If you have any doubts regarding the problem solving session itself, you can write it in the forum that is there in the NPTEL Swayam courses. And if you have any doubts regarding what I'm taking right now, the questions, you can either write it in the chat box or you can unmute yourself and tell me. Okay. All right. So I'll start, I think. So this is the second week of the NPTEL course Bioreactors. And before I start, I want to just give an overview of what we've done and briefly. So some we learned some important concepts regarding the growth rate of cells and how various derivations and calculations can be done regarding that. How to calculate in your bioreactors or in your reactions, how to calculate the rate of growth of the cells. Okay, and then we learned about the various models which help us calculate this growth rate, a few easy models, a few difficult models, and depending on the sort of reaction parameters, how many limiting substrates are there, or if there's an inhibitor, etc. Right? And then we also learned about enzyme reactions and their kinetics. And after we've done the basics of that, we learned about how enzyme kinetics is different when an inhibitor is involved, how the parameters of the enzyme reaction changes, and the different kind of inhibitions that are also possible. And the last module that you had this week was measurement methods, in which you can measure the growth of cells or the viability of cells, right? So we'll just go on to the questions that I prepared. So the first question is, which of the following are the products of a bioprocess? Remember in the starting, sir talked about what products of a bioprocess can be. So this question is regarding that. So the options are biomass, enzymes, biodiesel, and biopharmaceuticals. I'll just like to remind you all that the answer to these questions can be multiple also. So try guessing which is it. You can write the answer in the chat. Which of these is the correct answer? No one knows the answer? You can write it in the chat box, or you can unmute yourself and tell me also. I'm audible, right? I hope I'm audible.
Ma'am, all are possible answers of product of bioprocess.
Okay. Anyone else wants to answer? Like if you think all of the above is not the answer and it's something else, you can just write it in the chat box if you want. Alright. No answers as such. Alright. All C and D, all are possible products. C. Yeah, Raghav, I have started recording. Don't worry. These will be shared with you. C and D. The answer is technically all of the above because biomass is the cells. You can have reactions in which the end product that you require is the cell, right? And the enzymes could be the end product. A biodiesel, like we talked about it in the last session, could be the end product, and biopharmaceuticals like monoclonal antibodies could be the bioproduct, right? Okay. No one has a doubt in this, right? Then I'll just move on. In case you have a doubt, just please mention it in the chat and or write it or talk to me like by unmuting yourself. So question 2 is identify from below the logistic equation giving the cell growth rate. So these are the four options. Um, what do you think is the answer? Okay. A. This was mentioned by Sir in his lecture. You might not remember it, but I'll help you figure out what it is. B. Okay. Alright. Any other answer? Anyone? B. Okay. B. Do you guys know what a logistic equation is exactly? A yes or no in the chat will suffice. No. So, what exactly is the difference between an exponential or a logistic equation or growth in this term? So, the first equation that you alright. So, the first equation that you see in this option is the exponential equation, and the second that you see is the logistic equation. So, in this case, the answer will be B. So, what a logistic equation means is that in the first graph or the first sort of equation, you're assuming that there is no end point to the increase in population size. That over the time that the population can keep on increasing, which is not what we see in at least batch, right? In batch mode of operation, we don't see a continuous increase, right? So there is a point where the population will plateau, right? And that is what the logistic equation is trying to define. Okay. So in this case, the first equation is just assuming that as long as there is substrate and the optimum conditions given, the cells will keep increasing. Okay. And the second equation accounts for the fact that there is a maximum amount of growth possible by the cells. Okay. So that the population will plateau at some point. Plateau in this case means there will be no increase if you see it as a graph. Like there is equal amount of cells being formed and equally cells are dying also. So it will appear like there's a flat line on the graph. Um, were you guys able to understand that? Um, did you get
Yes,
that?
yes.
Yeah, all right. Okay. So okay, we can. Do you have any doubts in this question, or shall I move on to the next one? I'll just assume that doubts are not there and move on. All right. If you have doubt, just please unmute yourself. So the third question is, glucose is the preferred carbon source by microorganisms, and you know the reason because glucose is the starting. substrate in glycolysis, which is the energy deriving, which is one of the energy deriving metabolic cycles, right, in the cell, in all cells. Okay, but it is not preferred in industrial bioprocess. And the options for these are glucose is inexpensive, glucose is expensive, it does not require any pre-processing steps, and it is a preferred carbon source by microbes. So why do you think it is not preferred in industrial bioprocess? Glucose is expensive. All right. Sure. Okay. Any other answer? All right. Um, so I will tell you the answer. It is in fact the fact that glucose is expensive. So in cases like this, you tend to use things like starch and cellulose, which are readily accessible. You can just take it from a lot of food sources have starch, right? A lot of food sources, plant sources have cellulose. But to get pure forms of glucose, it's very difficult. You have to extract glucose by other enzymatic processes. Not a lot of organisms produce pure glucose. So it's that extraction process which will make getting glucose expensive. So instead of that, we directly use products which are able to give us glucose by some reaction, or that our microbe can directly use it because they have the enzymes to break down the cellulose or starch to give rise to glucose, which they can utilize, right? Um, Okay. Question number 4 will be the effect of concentration on the limiting substrate on the cell growth rate is given by the Monod model as, and one of these four options will be the correct answer. So in this case, limiting substrate is the substrate due to which the chances of the reaction stopping is possible. So when this substrate runs out, the growth also runs down. Okay. Or the product. In this case, if there are two substrates giving rise to one product, which is the substrate, the lack of which will cause, uh, uh, stopping of the reaction itself or slowing down of reaction itself. This also, sir, has explained. So, which is the correct answer? B. Okay. Any other answer? Let me see. B. Right. B. Most of you have, in fact, guessed the correct answer. It is, in fact, B. So this is the answer. This is the Monod model for uh, seeing the effect of the concentration of the limiting substrate on the cell growth rate, right? So question number 5 will be define Ks in the Monod model. So Ks is either the substrate concentration at which growth rate is half maximal. Ks is either the substrate concentration at which growth rate is equal to 1. Ks is the substrate concentration at which growth rate doubles. Or Ks is the growth rate at which substrate concentration does not have an effect on the growth rate. So what do you think is the answer? A. Okay. Anyone else has another answer? A. Yes. Okay. Okay. Most of you think it is A. And if you remember, this is the graphical representation of the Monod model, where the x-axis is the substrate and the y-axis is the growth rate. And you see it's a plateauing or a rectangular hyperbola, like Sir told. Okay. And this line is the maximal growth rate, after which you don't see an increase in the product or the cell concentration. And Ks is the substrate. Like in the Monod model, if Ks is equal to S, then you get 2S here and you can cancel it here so that you get this maximum growth rate by 2, right? So Ks is in fact the concentration at which growth rate is half maximum. The substrate concentration at which growth rate is half maximum. All of you guys have understood the graph, right? What the graph represents. right? Because changes in the reaction parameters will bring changes to these graphs and the Ks and the Vm, etc. Right? I'm assuming you guys have understood this. I'll move on to the next question. Okay. So question number 6 is that growth rate dependent products are also called. The options are secondary metabolite, limiting metabolite, primary metabolite, and high value metabolite. So the answer is primary metabolite. Okay. Any other answer? C, primary metabolite. Okay. C. Okay. So most of you seem to think it is C, and it is in fact C. So primary metabolites are metabolites that are required by the cell to stay alive. Okay. So things like enzymes of the glycolysis pathway will be primary metabolites. Secondary metabolites will be substances which are produced in specific cases. Like if the cell is under certain stress, then it produces a secondary metabolite which will help overcome it. Okay, and both of these could be our desired end product. So a lot of cancer drugs are actually secondary metabolites that the cell produces. Antibiotics are secondary metabolites. They are not needed for the cell to grow, but in case a bacteria is or a fungus is attacked by some other organism, it will produce these antibiotics to fight it off, right? So they will be secondary metabolites. All right. I'll move on in case you guys don't have a doubt. Okay. So question number 7 is, which of the following statement or statements are true with respect to substrate utilization under low level of substrate? So in case um, the cells don't have enough substrate to survive, like if you require 10 moles of glucose for your reaction, but you've only given 5, and that is not enough for the cells. The amount of cells that are there in the reaction or the bioreactor, what do you think will happen? The cell does not use the available substrate. The substrate goes completely towards cell multiplication. The substrate is utilized only for product formation, or the substrate is utilized for cell maintenance activity. Alright, so you think it is D. Any other answer? C and D. Okay. Right. Since no other C, right? Okay. Alright. So the answer is D. Substrate is utilized for cell maintenance activities. So the first option can't be correct because the cell cannot not use the substrate given to it. It will not stay in a dormant condition, right? And the substrate cannot completely go towards cell multiplication, because for cell multiplication also the cells need energy, right? And that energy is got given to the cells by utilizing the substrate so that they produce ATP molecules. Okay, so this option is also not correct. So C is substrate is utilized only for product formation. So depending on what the product is, this can be true or cannot be true. In case it is a primary metabolite, yes, the substrate will be produced. The substrate will be used for product formation, but that is not the only thing. So it is not only for product formation. So it will be used for cell maintenance activity, a byproduct of which could be the product that you require. Alright. So in case there are no doubts, I will move on to that is why this C and D option was not exactly right. So a better option will be D in this case, right? Okay. So question number 8 is, match the following inhibition types in A according to the binding of inhibitor in B. So like Sir has taught us, there are three types of inhibitions: competitive inhibitions, non-competitive inhibitions, and uncompetitive inhibitions, and they are differentiated based on where the inhibitor is binding, right? So that is what you have to choose in this case. So what do you think is the answer? C. Okay, fine. Alright. So the answer is, before that, just sort of let's recap what inhibitor is, right? So in this, what you can see in the first um, picture is the sorry, the first picture is the fact that the substrate is binding to the enzyme, right? That is called normal binding. In competitive binding, the inhibitor binds to the part of the enzyme where the substrate was supposed to bind, and the substrate cannot bind. So that is competitive inhibition because the inhibitor is competing with the substrate. A non-competitive inhibitor will be where the substrate will go and attach to a separate site of the enzyme, not the site where substrates usually attach, but in doing so, it will change the structure of the um, allosteric non-allosteric site, right? The site where the substrate is supposed to go and bind. And because the shape of the site has changed, the substrate can no longer bind. An uncompetitive inhibitor will be the one in which the substrate, the inhibitor binds to the substrate. Okay, and that is what the answer is in fact. So the C option. One will be Y, which is competitive inhibitor binds to the enzyme alone. Two is Z, where non-competitive inhibitor binds to both enzyme and the enzyme substrate complex, and uncompetitive inhibitor binds to the enzyme substrate complex alone. Right? Anyone has any doubts regarding this? If not, I will move on. Alright. So, question number 9 is, which of the following statements are true? So, Km is modified in competitive inhibition. Both Km and Vm are modified in non-competitive inhibition. Both Km and Vm are modified in uncompetitive inhibition, and only Km is modified in non-competitive inhibition. This you might not get from the top of your head, but does anyone know what the answer could be? If you want, I can go back to the model that I showed, the picture that I showed, or A and C. Alright, the answer is A and C. Sure. A and C. Okay. C, A and C. Okay. Alright. So the majority seems to think it is A and C. And let's see if the answer is correct first. And yes, A and C is the correct answer. And that is because in competitive inhibition, the red line in these indicate when there is an inhibitor, and the blue line indicates when there is no inhibitor, right? So you can see that in competitive inhibition, the slope increases, right? And the uh, the intersect is the same, right? So Km is increased and Vmax is unaffected. In case of uncompetitive inhibition, you see that it is the intersect itself changes, but the slope remains the same. So Km is reduced and Vmax is also reduced. Okay, and in non-competitive inhibition, you can see that uh, the Km is unaffected and the Vmax is reduced. Okay, so the correct answer was Km is modified in competitive inhibition, which is the first one, and both Km and Vm are modified in uncompetitive inhibition, which is the second one. Right? Moving on to the next question, in case no one has any doubt. So question number 10 is, which of the following is or are the suitable methods to estimate the viable cell concentration? Viable cells are also live cells or cells which are capable of either forming your product or multiplying so that you get a large number of mass of cells that you require, right? So the answer is dry weight, packed cell volume, dye exclusion, or plating. So what do you think is the correct answer? A, okay. C and D, okay. A and D, C and D, okay. Some same answer, some varying answers. Alright. The answer is, in fact, dye exclusion and plating. So C and D is the correct answer. So the reason why dry weight and packed cell volume cannot be used is in that you're taking the total mass that is present in your reaction. So you can't differentiate whether the cells are alive or not, right? So even dead cells will have a weight, will have a mass, and they will collect and they will add to the mass, right? That you're estimating by either dry weight or packed cell volume. Okay. But dye exclusion is because dyes like trypan blue are not absorbed by the cells. Okay, only dead cells which have cracks in the cell walls will be able to take in those dyes. So using that you can see how many cells are alive. And plating is because you're taking the solution or the reaction mixture and plating it on agar plate, only the cells which are alive will be able to form colonies. So if you count the number of colonies, you will be able to tell, and if you back calculate to see how many uh, are alive in the original solution, you will be able to tell how many viable cells were there, right? Because only alive cells will be able to form colonies. Dead cells will not form colonies and will not appear on your plate at all. Right? So in case no one has any doubts, anyone has any doubt? Okay, so the next question is, which of the following is an assumption made to obtain the Michaelis-Menten enzyme kinetics? So a certain assumption has to be made so that we get the formula that we got. So what is it? So the options are Km is dependent on the enzyme concentration. The concentration of enzyme substrate complex does not change. There is no free enzyme in the system and there is an accumulation of the enzyme substrate complex. So what do you think is the answer in this case? C and D. Okay. Anyone else? Any other answer? Or are you not sure about the answer in this case? Only C and D. Okay. Two people have answered. Alright. So let's see what the answer is. So the answer is B. The concentration of enzyme substrate complex does not change. And this graph will show you why. Alright. Because we are assuming that at time zero, no enzyme is attached to the substrate, right? But as soon as the time goes from above zero, most of the enzymes will at one point be attached to some substrate. Okay, and after that, at most points, the enzyme will be bound to some substrate. Even if if we're taking the example of one enzyme, it has attached to substrate, form the product and release the product. Again, it will attach to a substrate, right? So at a majority of the time till we get the maximum amount of product, the enzyme is in fact in complex with the substrate. It is not talking about the same substrate. It's not like the enzyme substrate complex is one and it does not dissociate. It's just that if you take the overall reaction, an enzyme is bound to a substrate, not the same substrate necessarily throughout a substrate, right? So the answer is B. The concentration of enzyme substrate complex does not change. Moving on to question number 12. Okay, so this is a calculation question. I hope you guys have a calculator and a laptop in which you have Excel in this in case you want to follow along. So consider an enzyme substrate reaction following micromomentum kinetics with the kinetic data given below. So what they have given you is the time in minutes, the substrate concentration in molar, and the velocity in molar per minute, right? And so you have to calculate the kinetic parameters Km and Vm. Okay, and these are the options given to you. Do you know how to do this? Can you think of how to do this? If anyone does know, Can you unmute yourself and tell? You don't know how to. Does anyone know how to do this?
Ma'am, we
1/x versus 1/v.
Okay, yes. So after you get 1 by m, 1 by vm and 1 by, oh sorry, 1 by s
I
and
mean,
1 by v, yes, sorry. What do
yes,
you do with that, um, data?
We need to
ma'am.
find slope and intercept.
Yes, all right. Do you guys know how to find this in Excel? If if
Yes,
anyone
ma'am.
doesn't, then just write it in the chat. I'll just show you how it's done. In case someone doesn't know. Okay. I'll just wait for one more minute in case someone's typing. Alright. So no one said anything, so I'm assuming all of you know how to do that. So I'll just show you. This is the graph, right, that you get from plotting 1 by v against 1 by s, right? So this is the line, this is the intersect, and this is the slope, right? So the slope in this case, right? So here you can see it is the the equation for the line, the best fit line, because this line, if you actually connect the points, it's not a straight line. So the best fit line will be one in which it shows what a possible straight line will be, right? And the equation for that will be in the form of y equals to mx plus c. Okay, where mx is also where mx is the slope and c is the intercept, right? And mx, which is also the slope, is equal to km by vm, like you can see here, right? Km by vmax is the slope, and the c uh, component that you get from this formula is the intersect, which is also 1 by vm. In this case, this is 1 by vm, right? So from when you the data given to you is in terms of S and V. So once you divide these individual um, data points, if you divide 1 by these individual data points in both cases, you will get this data. So you will get 1 by S and 1 by V. And if you plot a graph on Excel, this is what you will get, right? And the you will get an equation in this form. Okay. So in this case, the equation was y equals to 0.45x plus 1.089. Okay. So the slope is 0.45 and the intersect is 1.089, right? So, like I said, the intersect is 1 by Vm, right? So, since I just told you the intersect, the C that you got from the Excel sheet is 1.09. So that means 1 by Vm is equal to 1.09, right? So from that, if you rearrange this equation, you can find out that Vm is equal to 0.917. That is the calculation that I am getting, all right? And you know that the slope is equal to km by vm, right? And from the equation that I got, it is equal to 0.46. So again, if you rearrange the equation, and now since we have found out the vm, which is 0.917, you rearrange the equation and you divide 0.46 by 0.917, you get a value of 0.505, which is what I got, right? Depending on the software that you use to get this graph, this formula, the y equals to mx plus c that you get, could be slightly different. Okay, so there might be differences in your answer. So the option that is closest to the one I got is D, right? Where Vm equals to 0.918, which is almost what I got, 0.917, and K is equal to 0.419, which is almost equal to the 0.505 that I got. No other value is coming close. You also have to see if the um, you know units are correct or not. So in your data, the unit for substrate was given in M, and that is what is there in km, and your V was given by M per minute, and that is what you got in Vm also, right? There are some in which they are not correctly written, right? There could be some in which they are not correctly written, okay? So you have to make sure of that too. In this case, all of that is correct, but yeah, here millimolar is written, right, which is not correct, right? In few of the options, millimolar is written, right? That is not correct. All right. Does anyone have a doubt in this question? Were you able to understand how I solved this? Yes, got it. Okay. Subha, were you able to get how this is done, or do you want me to explain it once more? Yes, you understood it, right? And I'm assuming everyone has understood. If you have not, just write it in the chat. I'll explain it once more. Okay. So, question number 13, and this is the last question for today, is given the above data that we just had, what happens to the value of Vmax if the enzyme concentration in the system is doubled? So, in this, you're using, you know, amount of enzyme, right? So, if the enzyme amount was doubled, what do you think will happen to these parameters? So, what will happen to Vm? Okay, and the options are the value of Vm is half, the value of Vm is doubled, the value of Vm is unaffected, and the value of Vm equals to Vmax by 2. So what do you think is the answer? Unaffected, okay. Maybe it will help if I show you what the formula for Vm is. So this is the formula for Vm. Vm equals to K3 by K3 into Et. That is the total amount of enzyme. So K3 is the rate constant at that point, from where enzyme substrate complex, you know, is it goes into enzyme and product. So now, do you think it is unaffected? Any other answer? Anyone else? Alright. No one else. No one else thinks it is anything else. Not A, B, or D. B. Okay. Alright. B. The answer is in fact B, because you can see Vm is directly proportional to the enzyme, total enzyme, right? So if enzyme is doubled, then Vm will also be doubled. So Vm is the correct answer. So that is all the questions that I have for today. If you guys have any doubts regarding any of the questions, I can go back and take it once more. Do you guys have any doubts, or any other doubts regarding the module that you took for this week? Okay. So, since none of you seem to have a doubt, how to find wait, how to find limiting reactant? Okay. So, in that case, like the equation that you remember, just one second. I this okay. I'll just show you in another slide maybe. I'll stop sharing this slide and I will take a normal PPT. Just a second, okay? Hope my PPT is visible, right? Just can someone
Yes,
confirm?
ma'am.
Right? Okay.
Yes, ma'am.
Okay. So, like Sir gave an example of A plus plus B, B is equal to maybe 2 moles of the product, right? Should I increase the font size to a little bit more? Right. You can see this, right?
Yes, ma'am.
Okay. So he had given an example in which 10 moles of A was there and 10, 10 moles of B was there in the reaction. Like you have supplied 10 moles of A and you have supplied 10 moles of B. Okay. Okay. So how do you find the limiting substrate in this? Anyone has any guesses on what it is? Before I begin, I just like to tell you that limiting substrate is not because of its property. It is because of the amount that you put it in. Okay. That is why it's limiting. It's not because it has an inherent property which is limiting. The reactant which forms least product. So not really. So what this equation means, the first equation that you saw, this A plus 3B equals to 2P, where P is the product and A is reactant 1 and B is reactant 2, right? So that means you need 1 mole of A and 3 moles of B to produce 2 moles of product. This is how the reaction works. Okay, so you know any chemical reaction will be balanced, right? So you don't always add equal amounts of all reactants and you won't no chemical like most chemical reactions don't happen like that. So just like that, in this case, A has to react with 3 moles of B. 1 mole of A has to react with 3 moles of B to form 2 moles of product. There is no other way. Okay, this is a balanced reaction. So in case you had given equal amounts of A and B, so that means you have 10 A, right? So if you had to get uh, full amount of product from the A that you had, right? So you will need 3 times that of B. So you will need 30 moles of B, right? 30 moles of B, right? 30 moles of A. Does that make sense? Can someone say yes or no?
Yes, ma'am.
Okay. So uh, to get in this case, if you had 10 moles of A, you would have needed 30 moles of B to get this 2 moles of product. But since you have only put 10 moles of B, you will only get a little amount of product. You won't get 10 moles worth of the product. Sorry, in this case, 20 moles worth of products, right? You won't get that. So in this case, B is the limiting substrate because you don't have enough of B, right? So you will have um, you will have A left over. You will have A left over because there is not enough B, and for the reaction to be completely, completely formed. Do you get that in this case? So just let's try another thing. So maybe A plus 2B plus C, C is giving you product. Doesn't matter how much moles of product in this case. So and you have um, seven, maybe about 10 moles of A, okay, and you have 30 moles of or maybe, yeah, maybe B, and you have 30 moles of C. So in this case, what do you think is limiting? 30 moles of A. Like for the reaction to, like your whatever initial substrate you have given, for it to be completely formed into a product, what do you think you will need to add more of? Does anyone know the answer? If not, then I'll explain once more. Can anyone guess what it will be? What is limiting in this case, and what is less in this reaction? Okay. What will be left over even after some product is formed, because the reaction cannot go further. That is your limiting substrate. C. Okay. So you think it is C. Okay. It's actually B, sorry, A and C both, right? So in this case, think that what is, how will you divide this, right? You will divide this by 2. So if you need 2 moles, so here 30 divided by 2 will be 15, right? Okay, so you're assuming that 1 mole is 15 in this case. 1 mole is 15. So 2 moles will be 30, right? Okay. So in this case, A also one, the moles will be same for everything, right? So in this case, 1 mole of A will also be 50. So you don't have enough of A also, right? And you don't have enough of C. So 15 into 3, okay, that is 45. You would require 45 C and 15 A for this reaction to happen completely. But you have less of both A and C. Okay. Did you get how it is done? Anyone? Yes, ma'am. Anyone who didn't get it, just write that in the chat. If there's anyone who didn't get it, we can take one more example if you want. Understood? Understood, everyone? Is there anyone who's not understood? Then just write that in the chat, or you can unmute yourself. I'm assuming you have understood. Okay. So I think that's all for today. I hope you guys understood what I was trying to tell you, and hope your doubts are clear. We'll meet again the next week for module 3, problem solving. I hope you guys are able to solve the assignment that is there right now. You still have a little bit of time, I think. Please show all answers. For for what exactly? Vijay, can you tell me what answers you want to see? Vijay Aditya C. All right. Oh, so you came late. All right. So uh, all this is the end for the session. Just wait, Vijay. Okay, I'll show you the questions once more. Those who have attended can leave the meeting if you want. I'm just going to repeat it once for Vijay. I'll see you guys next week. So bye. Those who have any doubts can stay back. Thank you everyone for joining. Apart from Vijay, are the people left the ones who have doubts? Alright. Um, okay, Vijay, I'll just show you the questions once more. Let's just go through it. Alright, just present it. I hope my screen is visible again. Right. Okay. So this is mostly for Vijay and anyone else who just wants the recap. So Uh, this is just an overview. So you learned in this week topics about how to determine the growth rate of cells, the various models which will help you calculate that. What are enzyme reactions and what are its kinetics? How enzyme kinetics um, is different when an inhibitor is involved, and different measurement methods, right? So question number one is, which of the following are products of a bioprocess? The options are biomass, enzymes, biodiesel, and biopharmaceuticals. Right? Uh, Vijay, do you want to try guessing what the answer is?
Biomass, biodiesel and biopharmaceuticals.
सुनाई नहीं दिया आप चैट पे लिख दीजिए प्लीज A, B, C, D. Okay. You're almost correct. Technically, all of them are the correct answer, right? Because, like Sir told at the start of the module, that your end product could be cells, which would be biomass, or it could be biodiesel, like the last time in the last module he told us about biodiesel. It could be enzymes, or it could be biopharmaceuticals like monoclonal antibodies, right? So, question number two: from the below equation, what is a logistic equation for giving cell growth rate? So, do you want to guess the answer? C. Okay. The answer is actually B. Okay. So I'll just explain what a logistic equation is. So the first equation that you see, where Rx equals to nu x, is an equation for exponential growth. So that means we're assuming that how much ever substrate we give, the cells will keep on increasing. Okay. There won't be a time where cells will not keep on increasing. So you can increase it to infinity. Okay. But that is not the case in logistic growth. So in logistic growth, we're assuming that at one point the cells will sort of plateau. Okay, so that means there will be a time where no more increase in cell growth, or at least apparent cell growth will take place. Cells will still be dividing, but the thing is equal amounts will be dying. Okay, so yes, Matheshwaran, you had a doubt. I hope I pronounced it correctly. You raised your hand. Do you have a doubt?
No, ma'am.
Okay. So where was I? Yeah. So that is what a logistic growth means, where apart from the factor for the exponential part, we are also incorporating a sort of decline phase of where the cells will stop growing. So that is why the option is B. Okay. That is the logistic equation. Okay. So question number three is, glucose is the preferred carbon source by microorganisms, but it is not preferred in industrial bioprocess. And the options are, it's either glucose is inexpensive, glucose is expensive, it does not require any pre-processing steps, or it is the preferred carbon source by microbes. What do you think the answer is? D. Okay. Actually, the answer is D, because um, it is actually preferred source by microbes, but that is not the reason why it is not used in industrial bioprocess, right? So if we had the option, we would actually use this, right? Because it is preferred by microbes. But what they're asking is, it is not preferred in industrial bioprocess, where our main, cells are microbial, and that's because glucose is expensive. So if you have to have reactions where there are 20 liters to the reaction mixture, you can't use glucose as a substrate because that will be very expensive for you. So that is why option is B. Fourth is the effect of concentration on the limiting substrate on the cell growth rate is given by Monod's model as. So what do you think is the answer in this case? B. Okay. Yeah. So that is the Monod's model, right? So B is the correct one. So define Ks in the Monod model. So what does Ks actually signify? So Ks is either the substrate concentration at which growth rate is half maximum. Ks is the substrate concentration at which growth rate is equal to 1. Ks is the substrate concentration at which growth rate doubles, and Ks is the growth rate at which substrate concentration does not have any effect on the growth. So what do both of you think is the answer? Okay. Any other answer? C and D. Okay. The answer is actually A. Okay. So this is the graph which graphically represents this model, right? So Ks is what you see here. Okay. And that is Ks is, you know, um, the at if Ks you assume is S is equal to S, that is the substrate concentration is equal to Ks, what you get is S plus S, right? That is 2S, and there you can cut the S from the numerator and the denominator, so you will get a 2 here. So in that case, the growth rate will be equal to the half of the maximum growth rate, right? Maximum growth. Okay, that is Ks. So at what point the amount of growth is half is exactly Ks. So that is the substrate concentration at which growth rate is half maximum, is half of the maximum growth rate which is possible. Okay, I hope you understood. If you do have doubts, please do write it in the chat. I'll move on to the next question. Okay. So question number 6 is growth dependent products are also called secondary metabolites, limiting metabolites, primary metabolites, or high value metabolites. What do you think the answer is? C. Okay. A. C. Okay. The answer is C. So secondary metabolites are products that are formed by the cell when a particular sort of environment it is exposed to. Okay. So, for an example, like Sir taught you about glycolysis, right? In which glucose is converted to pyruvate. It is one of the very primary metabolic, cycles that are there in all cells. Almost. That is the system through which we produce energy, right? So any enzyme which is present in that system is a primary metabolite. You require it for the cells to live. Okay. Secondary metabolites, an example would be antibiotics. So penicillium does not normally produce antibiotics, but when it is exposed, like normally does not produce high levels of the antibiotics, but when it is exposed to something that is attacking it, it will produce antibiotics, okay, which will help it fight against these invaders, which could be bacteria. Okay, so that is an example of secondary metabolites. So growth dependent products, that is what is there with just the growth of the cells, is primary metabolites. Okay, I'll move on to the next question. Question number 7. Which of the following statements are true with respect to substrate utilization under low level of substrate? So, if in your reaction you have about 100 cells and they require maybe 100 kgs of glucose, but you've only given them maybe um, 50. So what happens? Does the cell not use any substrate, or the substrate goes completely towards cell multiplication, or the substrate is utilized only for product formation, or the substrate is utilized for cell maintenance activity? What do you think it is? B. Okay. C and D. Okay. The answer is D. Substrate is utilized only for cell maintenance activity. That is just to be able to keep the cell alive. Okay. So the cell will not, the cell will only multiply when it senses that there is enough, nutrients for it to divide into two and for both of those cells also to be alive. Otherwise, it's a waste, right? If it divides into two, but then there is not enough nutrient for both of those cells to survive. Okay, so the answer is substrate is utilized for cell maintenance activities. Okay, and it's not like the cell cannot use the available substrate; it can't just die off, right? That is why. So, question number 8 is match the following. Inhibition type A according to the binding of inhibitor in B. So, there are three types of inhibition type, right? Competitive inhibition, non-competitive inhibition, and uncompetitive inhibition, depending on where the inhibitor is binding, right? So, what do you think the answer to this is? C, C, okay. Right. The answer is C. Okay. So in normal binding, the substrate will go and bind to the enzyme. In competitive inhibition, the inhibitor will bind to the site where the substrate was supposed to bind. And since now it is occupying that site, the substrate can't come and bind. So it's competitive inhibition. In non-competitive inhibition, the inhibitor will go and bind in a separate site, but that will change the configuration of the site where the substrate was supposed to bind. And because now the configuration has changed, the substrate cannot bind. And in uncompetitive inhibition, the inhibitor will bind to the enzyme substrate complex, and but it will not allow the product to be formed. Okay. So the answer is C, where 1 equals to Y, that is competitive inhibition is where inhibitors bind to the enzyme alone. 2 is Z, non-competitive inhibitors bind to both enzyme and enzyme substrate complexes, and uncompetitive inhibitors are the ones that bind to the enzyme substrate complex alone. Okay. The question number 9 is, which of the following statements are true? Km is modified in competitive inhibition. Both Km and Vm are modified in non-competitive inhibition. Both Km and Vm are modified in uncompetitive inhibition, and only Km is modified in non-competitive inhibition. So, what do you think the answer is? C. Okay. C. Okay. So the answer, like I'll just show you the Lineweaver plots for these. So in competitive inhibition, what you see is that the intersect remains the same, where the line will intersect in the y-axis will remain the same, but the slope has changed, right? So in that case, in competitive inhibition, Km has increased and Vmax remains unaffected, okay? Because 1 by Vmax is dependent on the intersect, right? And since that is not changing, Vmax is unaffected, but since the slope is the Km, you find Km through the slope, and since that is changing, the Km will have increased. Okay, so in uncompetitive inhibition, what you see is the intersect itself has changed, but the slope has remained the same. So Vmax is reduced and the Km is reduced because the formula for slope is Km by Vm, right? So even if one of those things changes, the slope will change, right? So in this case, Km is also reduced and Vmax is also reduced. Sorry, and in non-competitive inhibition, you see that uh, this sort of thing happens, right? So what happens is Km is unaffected because the slope, both of these things have changed. The slope has also changed, the intersect has also changed, but in this Km remains unaffected and the Vmax is reduced. Okay, so the answer is Km is modified in competitive inhibition. Yes, Km is in fact modified in competitive inhibition, and both Km and Vm are modified in uncompetitive inhibition, right? Question number 10 is which of the following are suitable methods to estimate viable cell concentration, which are live cells, okay? So the options are dry weight, packed cell volume, dye exclusion, and plating. So what do you think the answer is? B. Okay. There could be multiple answers. A, B, C, D, everything, and D. So the answer is actually diffusion and plating. So what viable cells mean, like I told you, is that they're live cells, right? So imagine you have a mixture of live cells and dead cells, and you take the dry weight of that. How will you differentiate which is live and which is dead? And the same thing happens with packed cell volume, right? You don't know which is live, which is dead. But in dye exclusion, some cells, which are the dead cells, are able to take in dyes like trypanosine, but live cells do not take them in. So if you put them under a microscope and you count how many are colored and how many are not colored, from the total amount of cells you'll be able to tell how many are alive. And in plating, what you're doing is taking a little bit of the solution, reaction solution, and plating it on agar plate. Only live cells will divide to give rise to a colony. Even if you have plated dead cells, they will not grow. So what you will be able to observe is the colonies of live cells. So if you back calculate, you'll be able to get in your original solutions how many live cells were there. So the answer is C and D. Okay. So question number 11 is which of the following is an assumption made to obtain the Michaelis-Menten enzyme kinetics? So what is the assumption? It's either Km is dependent on the enzyme concentration. The concentration of enzyme substrate complex does not change. There is no free enzyme in the system, or there is no accumulation of enzyme substrate complex. So what do you think the answer is? B. Okay. B and C. Okay. Any other answer? B and B and C, right? So the answer is B. So the concentration of the enzyme substrate complex does not change. Okay. So for this Michaelis-Menten equation, you are assuming that after a point type t, time t, where at time t equals to 0, no enzyme would have been, forming enzyme substrate complex. But as soon as the reaction starts, right, what you see is that all the enzymes will be saturated by the substrate if there is enough substrate for the reaction to take place. Okay, and throughout the reaction, the amount of enzyme substrate complexes will almost be equal. And it's not saying that the enzyme once an enzyme has formed a complex with the substrate, it will not, you know, dissociate. It's not saying that. It's just saying that once it has dissociated by giving rise to a product, another molecule of substrate would have come and bound to it, right? So if you see the average, it will look like the enzyme substrate complex is remaining the same. So that is the assumption that you have to make. Okay. So question number 12 is calculation. So consider an enzyme substrate reaction following the Michaelis-Menten kinetic. The kinetic data given below. So time points have been given. substrate have been given and the velocity of the reaction has been given, right? And you have to find the kinetic parameters Km and Vm. Okay, so how do you do that? I'll just explain that to you. So this is the line of Brewer's graph, right? This is the graph where you have, this is what the graph you get after plotting 1 by V and 1 by S, right? That is the substrate and velocity, right? So the line that you get, you the slope will be equal to Km by Vmax and the intersect will be equal to 1 by Vmax, right? So to find the Vmax and the Km, what you have to do is, since you have been given the substrate concentration and the velocity, you will divide 1 by V. So you will do 1 by S and you will do 1 by V and you will get the following data. And then you will plot a graph. This is the line you will plot, right? And you will find the best fit. So you see the points that are there don't actually form a straight line if you plot them, right? So you find the best fit which will give you a straight line, possible straight line, and then you see if you see to give the equation for this best fit, it will give you that if you do it in Excel, okay? And that equation will be in the form of y equals to mx plus c. So after I did this in Excel, the equation I got was y equals to 0.4x plus 1.089. Okay, so that is the equation in the form of y equals to mx plus c, right? So we know that mx is equal to the slope and slope is equal to km by vm, right? And c is the intercept which is also equal to 1 by vm, right? So you know 1 by Vm is equal to 1.09. So if you rearrange the equation, you get to see that Vm is equal to 0.917, right? And you know Km by Vm equals to 0.46 from your equation, right? So again, if you rearrange it, you will get Km if you divide 0.46 by Vm, which you just calculated. So if you divide 0.46 by 0.917, you'll get a value of 0.505. The nearest answer in this case is D. Okay. So where Km equals to 0.419 and Vm equals to 0.918, which is what nearly what I got, right? Okay. Does anyone have a doubt with this? How we calculated this? You do please
No,
either
ma'am.
no or yes. I didn't hear properly. Sorry. If it is yes, you have a doubt, just type it in the chat box. That is easier for me to know, ma'am. Okay. All right. So the next question is, given the above data, what will happen to the value of Vm if the enzyme concentration in the system is doubled? Okay. So what happens to Vm in case if you had taken maybe 10 moles of the enzyme, what happens if you now take 20 moles of the enzyme? Okay. So does Vm get halved? Does Vm double? Is Vm unaffected or does Vm become equal to Vmax by 2? So what do you think the answer is? So this thing B, D. Okay. So the answer is, this is the formula for Vm, right? Vm is equal to velocity constant K3 by total amount of enzyme. So you can see it's a direct relation, right? It's directly proportional, right? So if E is doubled, then Vm will also be doubled, and that is the answer. So value of Vm will be doubled. Okay. So that's all the questions that are there. Do you guys have any doubts? If not, uh, the session for today is finished. You can leave the meeting.
Ma'am,
Yeah,
I have a doubt.
yeah.
In limiting substrate, can you explain it with three reactants and the product will be 2P?
What? I did not get it. Sorry.
Limiting substrate, ma'am.
Okay,
Can you explain it with three reactants?
okay. I can try that. So do you know what a limiting substrate is? Can you try to explain what a limiting substrate is?
So it is the substrate in the medium that limits the growth.
Yes. So in the example that sir had given, do you know which one was the limiting substrate? Do you remember? He had given, I think, A plus 3B equals
Yeah,
to
ma'am.
product,
B.
right? B was the limiting substrate, right? So let's try with that first. A plus B, C, B equals to product, right? Any moles of product. Okay. So in this case, why is B the limiting substrate? Okay. So, and we had said that you have 10 moles of A and 10 moles of B, right? So in this case, so you have 10 moles of A, okay? And if you have 10 moles of A and you have to convert all 10 moles of A to product, you need 3 times that of B, right? So that means you need 10, you have 10 of A and you need 30 of B, but you only have 10 of B, right? Okay, so you won't be able to convert the entire substrate to product because you don't have enough of B. You needed 30, but you only have 10. Okay, so that is why B is the limiting substrate, right? So in case, imagine plus was maybe 2C, okay, and to give product, okay, and you had maybe 10 moles of A and
Ma'am, please explain it with 2P.
with
2P. The product will be 2.
why does it have to be 2P? Because it doesn't matter, right? I know that is, I think, a question in your assignment, right?
Ma'am, I have a doubt. Does it
Yeah,
affect the reactants, the product?
yeah. So that is a doubt, right? Just one second, I will explain it to you. Okay, I will change the equation. Change it to maybe 5. And here, let's see what happens is maybe 1.5. So in this case, imagine you have 10A, 10B, and 10C. Okay. Since A is the one which is used up completely in this reaction, it doesn't have anything associated with, we will take this as reference. Okay. So if you need the entire amount of substrate to be converted, you need the entire amount of A to be converted, right? So in this case, you have 10 moles of A. Okay. So what you have is 10 moles of A. If you have to have a perfect equation in which all 10 moles of A are converted to the product, then you need 7 times that of B, which will be 70 moles of B, right? And you will need 1.5 times, right? So that is 15 moles of C. So the only thing you have enough of here is A. So in this case, both B and C will be the limiting product. Did you get that?
Okay, ma'am.
Yes or no? The voice is not coming clearly. So can you write it in the chat? Yes. I'll stop sharing this. Anything else that you guys have a doubt with? Alright. Then if you don't have a doubt, you can leave this meeting. I'll also stop